eukaryotic cells. So both plant and animal cells would be eukaryotic cells. Remember how to spell that. Remember, if you have trouble, remember you have eukaryotic cells. Some people still mix up prokaryote versus eukaryote. So you have eukaryotic cells. You have cells that have membrane-bound organelles. Okay, so let's look here. This is our animal cell. Hopefully you are able to recognize that. Our animal cell over here, our plant cell over here. Okay, remember in plant versus animal cell, we've got a few things that are going to be a little bit different. Okay, so animal cells will not have a chloroplast. They don't need one. They don't do photosynthesis. No central vacuole, but they do have other vacuoles and vesicles, but not a central vacuole. In the tonoplast is the membrane. You can see it over here. The tonoplast is the membrane that surrounds the central vacuole. So obviously an animal cell wouldn't have a tonoplast. No cell wall and no plasmodesmata. And we're going to talk about what a plasmodesmata is near the end of this, but that's essentially a way that plant cells can communicate with one another. Plant cells, on the other hand, are going to be missing some things that animal cells have. Okay, down here you'll see that your plant cells aren't going to have lysosomes. They do digestive properties. They're not going to have centrioles. They're not going to have flagella, with the exception of some plant sperm. In particular, in our lesser plants, the ones that are not quite as evolved, like ferns and bryophytes, those that still require water to be able to reproduce. Those plant sperm will have a flagellum, so they can travel. So we're going to go kind of organelle by organelle and do a little bit of review. Like I said, this should mostly be reviewed from your freshman year, with a few things maybe added here and there. So let's take a look first here at the nucleus. Okay, our nucleus is going to contain a nucleolus. That's that dark spot right here. Okay, inside the nucleolus is going to be the DNA that is used to produce rRNA. Okay, so rRNA is ribosomal RNA. And so that's where you're going to find the DNA to produce the ribosomal RNA, is there in the nucleolus. Some other things that you will find in the nucleus would be our chromatin. If y'all remember, chromatin is DNA. Chromatin is DNA in the loose form. We usually think of chromosomes as our DNA, but our DNA does not condense into the chromosome form until where the cell is about to go through mitosis. In the very beginning of prophase, that chromatin will condense. So chromatin is this kind of loose spiderwebby looking part of DNA. Okay. Also inside the nucleus, you're going to find proteins. You'll find RNA. Remember, this is where transcription is going to happen. So you'll find the messenger RNA. The nucleus is also going to be surrounded by a nuclear envelope. If you come look down here, you can see this nuclear envelope. It's a double membrane here, okay, and it's got these pores in it, these little pores or holes, okay, and these pores are too small for DNA to fit through. That's part of why, again, remember, transcription is DNA to messenger RNA. DNA is unable to leave the nucleus because it can't fit through these holes, but the messenger RNA can. So the nuclear envelope is going to surround the nucleus, which is going to contain the nucleolus with the DNA for the rRNA. It's going to contain, the nucleus will contain the chromatin, that DNA. It'll contain proteins. It'll contain RNA. The nucleus, the nuclear membrane in particular, is also continuous with our endoplasmic reticulum. Okay, there's your spelling of endoplasmic reticulum. But you can see here how the nuclear envelope is continuous with that endoplasmic reticulum. And the endoplasmic reticulum, if y'all remember, has two parts. Okay, you have a rough endoplasmic reticulum and you have a smooth endoplasmic reticulum. Remember, the difference between rough versus smooth was ribosomes. Rough endoplasmic reticulum has ribosomes attached to it when proteins are being synthesized. And so the rough endoplasmic reticulum can help modify proteins, and then it's going to send them to the Golgi apparatus. So they can either be exported or incorporated into the membrane. So your rough endoplasmic reticulum is going to help modify these proteins, okay, that are being made right here with these ribosomes that are associated with it. And then those proteins that have been modified by the rough endoplasmic reticulum will head to the Golgi apparatus, or to become part of the cell membrane. The smooth endoplasmic reticulum, you'll notice it has no ribosomes. Okay, so there are no ribosomes there. I don't even know what I'm doing here. Ignore my writings. So there are no ribosomes on the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is going to help with lipid synthesis, in particular phospholipids. So the smooth endoplasmic reticulum is going to help make phospholipids. It's going to help break down glycogen. If you guys remember what glycogen is, hopefully you do. Okay, glycogen is the storage form of glucose in animal cells. So it's going to help break that glycogen down into glucose. And it's also going to help with detoxification. Okay, so it's going to take poisons or toxins, and it's going to add alcohol groups to them. Remember your OH there? It's going to add an alcohol functional group to them, which will make them a little bit more soluble so they can be broken down easier. Also in muscle cells, there's a modified version of the smooth endoplasmic reticulum that's going to help store calcium. Calcium is necessary for muscle contraction, and so it's called the sarcoplasmic reticulum in muscle cells, will store the calcium. So rough endoplasmic reticulum is modification of proteins. Smooth endoplasmic reticulum makes phospholipids, breaks down glycogen, detoxification. In muscle cells will store calcium. So those proteins that are made in the rough endoplasmic reticulum, they're going to head over to the Golgi apparatus. Okay, Golgi apparatus, Golgi body, Golgi complex, it's all the same thing. Okay, oh, I'm writing on that. Sorry, I was trying to make that bigger. Okay, so let's take a look at this a little bit bigger. So on the right there in your black and white, you've got your, that's a microscope view, an electron microscope. You can see the various vesicles here, okay, going in, okay, and then coming out as they're being done being modified. Okay, so we've got two sides to the Golgi apparatus. The Golgi apparatus is polar. Okay, so it's made of these flattened membrane stacks, and it is polar. And so it has what's called a receiving side, a cis or receiving side, and then across from that would be the trans side. Remember, trans is across. So all of your vesicles will enter on one side, and they'll come out the other side. And when they come out the other side, they are going to have the proteins that were in there will be modified. And they will, the Golgi apparatus will also contribute to the cell membrane. Okay. In addition to continuing to modify proteins for them to be shipped out and contribute to the cell membrane, some of your polysaccharides, remember saccharide is sugar, some of your polysaccharides are made here in the Golgi apparatus as well. Lysosomes are, if y'all remember, your lysosomes, they lyse or break things apart. These are your digestive organelles. They contain those digestive enzymes. Their pH is usually very, very low, okay, because they contain hydrolytic enzymes. Okay, lysis, lytic, break things down with water. So you can see here with this cell, the food has been taken in through phagocytosis, remember cell eating. Okay, the lysosome will join with that food vacuole and release its enzymes, and that'll break the food down into smaller pieces. Okay, the lysosome can also break down, old, worn-out parts of the organelle. That's what's happening here, breaking down this little portion of the Golgi apparatus. Okay, but you'll see your lysosomes came from these modified proteins. Remember, enzymes are a kind of protein. And so here's my rough ER making the enzymes here. Okay, they're getting made and modified. They're headed to the cis side of the Golgi apparatus. They'll come back out the trans side, and you'll see they're a little bit different. They've been modified there, okay, and turned completely into the lysosome. Your vacuoles and vesicles are both for storage. Okay, so here we're talking also about vesicles. Really, their main difference is size. Your vacuoles, as a general rule, are larger than your vesicles. They'll store water. They will store nutrients, food. Remember, plant cells have that central vacuole. Like we said on that earlier slide, it's surrounded by a tonoplast. That's the membrane that surrounds the central vacuole of a plant cell. Okay. Animal cells, remember again, they have vacuoles. They're just not as large. Okay. They also have vesicles. Some of your freshwater protists will have what are called contractile vacuoles. Okay. So they do just what their name says. They're going to contract. Okay. And because they are freshwater protists, that means usually more water is coming into them, so they need to get water out of them to help maintain their water balance. And so these contractile vacuoles will act kind of like pumps to squeeze water out of them. So these organelles that we've been talking about consist of your endomembrane system, okay, your endomembranous system, also known as the cytomembrane system. Okay, and so these are the organelles that are going to have resulted from, if you remember back when we talked about endosymbiosis a little bit ago, you had this infolding of the plasma membrane here. Okay, and so as this plasma membrane infolded and compartmentalized, you developed these various organelles. Okay, your nucleus, your endoplasmic reticulum, Golgi apparatus. And so at some point in time, these organelles were either continuous with one another or they came from one another. So our main organelles that are part of this endomembranous system are going to be the nucleus, in particular the nuclear envelope, okay, the endoplasmic reticulum, so things move from the nucleus, again in particular the nuclear envelope, the endoplasmic reticulum, the Golgi, and then on to the plasma membrane. You can see the vesicle there fusing with the plasma membrane. So these, all these organelles that are part of the endomembranous system, they're all going to need to be made up of phospholipids, okay, which would lend support to the fact that this cell membrane has invaginated in to form these organelles, because the cell membrane is made of phospholipids. There's no way this vesicle here would be able to join with the cell membrane if it was not made of the same stuff. Okay, so these organelles used to be continuous with one another. Okay, next we have what are called microbodies. Okay, so microbodies, we've got two examples we're going to look at here. Microbodies basically contain enzymes for specialized functions. One we're going to look at here is a peroxisome. Okay, peroxisomes contain catalase. Hopefully by now you guys know what catalase does. Okay, catalase is that enzyme that helps break down hydrogen peroxide, because hydrogen peroxide is toxic to our cells. So peroxisomes help contain catalase that will help break down hydrogen peroxide. Glyoxysomes down here, okay, these convert fatty acids into sugars in plants. So these are found in plants, and again they're going to convert the fatty acids to sugars. Our next organelle is a mitochondrion. Okay, hopefully we remember what this does. We associate it with that powerhouse, the ATP formation. Okay, so mitochondrion, they are going to have, they're a double membrane structure. Okay, that inner membrane will be all folded into what are called cristae. Okay, and the benefit to having these folds on that inner membrane is that's going to increase the surface area. So the pathways, those enzymatic pathways that are used to make ATP, that allows for more of them to occur because we've increased the surface area. Next, we've got our plastids. Okay, we have two basic types of plastids. We have what's called a chromoplast. Okay, these are going to contain some kind of pigment, carotenoids. Chloroplast is a kind of chromoplast. It contains chlorophyll. They could contain xanthophylls. Okay, so these plastids, they are the two types we've got again are the chromoplast. Those are going to do photosynthesis. In particular, the chloroplast with its chlorophyll pigment. Okay, but these are our color plast here. We're going to talk about the structures, both of the mitochondria and the chloroplast, much more in depth when we do photosynthesis and respiration. But again, similar to the mitochondria, you can see there's a bunch of folds there in the chloroplast to help increase the surface area for these enzymatic reactions to happen. The leucoplast, okay, our other example here is a leucoplast. The leucoplast is going to store starch. Okay, so remember, starch is the storage form of glucose in plants. All right, now we've got our ribosome. Hopefully you guys remember what a ribosome does. Remember, ribosome does protein synthesis. Okay, ribosomes are going to be made of rRNA and proteins, and they will help take that messenger RNA and they will turn that messenger RNA into the amino acid chain, into the proteins. They are in charge of the primary structure of the protein. The ribosome has two subunits, a large and a small, that are separate from one another until the messenger RNA enters the cytoplasm, and then those two will join together around the